Okay, so thank you, Colin, for the introduction. So I will talk about cryo-EM model refinement, and I will start with a brief overview of cryo-EM maps and models, because this is important to know in order to make some decisions when we design the cryo-EM refinement, the real space refinement. So currently there are about 4,200 cryo-EM models deposited in the PDB. So I checked a couple of days ago. That's a quite recent number. And this graph here shows the experimental methods which have been used to determine determine structures that are deposited in the PDB. So the main method is X-ray diffraction with 89%, and that's followed by NMR, 8%, and finally we have cryo-EM with 3%. So cryo-EM is now the third major method to determine structures. It's also interesting to look at the growth. So here we have the number of cryo-EM models deposited per year, starting from 2005 and going to 2019. And in the first decade of the 2000s, we have about less than 50 structures per year. And then from about 2014, we have a very large increase. And now we have more than 1,000 structures per year. Now it's also interesting to look at some properties of these deposited models. So again, we have here number of models deposited per year. And this time I restricted the number to models which are larger than 1,000 kilodalton, so which are very large. Now the red bars represent X-ray structures and blue cryo-EM structures. And we can see that over time we have fewer large X-ray structures which have been deposited, and we have more large cryo-EM structures. So this means cryo-EM is used to determine large structures, and this is important for refinement. Then it's also interesting to look at the resolution of the maps. So here we have the deposited maps per year. Again, we can see that increase, which starts 5 years ago. And here we have the different colors represent different resolutions. So here in dark blue, we have map resolutions better than 3 angstrom. Here we have between 3 and 4. That violet here is between 4 and 5. And finally, we have a couple of maps with associated models which have lower than 5 angstrom resolution. So what we can see here is that the biggest growth is in the range of 3 to 4 angstrom. And we also have a substantial number of maps in the range of 4 to 5. But now in the last year, we also can see that we have more and more high resolution structures, maps. So before moving on to refinement, I'd like to contrast the workflow of the two methods, X-ray diffraction and cryo-EM. We've seen that a little bit already in some of the talks today. So for crystallography, when we prepare our sample, we want to get crystals. Once we have crystals, we move on and collect data. So we perform a diffraction experiment, and then we get images, and we process these images, and this will yield structure factor amplitudes. So we all know that this is not enough to calculate an electron density map, so we have to get phases. Then once we have a map, we can build an initial model, and we can perform refinement. So for crystallography, refinement is often or most often done in reciprocal space. And so then we iteratively perform refinement and validation in order to get the best possible model. Now for cryo-EM, the workflow is very similar. So first we have to prepare our sample. We collect data on an electron microscope. We get images, and we process these images in order to get a 3D map. So here we don't have this procedure of phasing. So with that 3D map, we can build a first model, then we perform refinement in real space, and again, we do validation and refinement iteratively. So now what is refinement? So here is one definition. I took this definition from a paper from the Electron. Of course, there are many other definitions. I just chose this one because the associated paper is actually quite a nice introduction to refinement. So for those who are not so familiar to that. So in this definition, refinement is the optimization of a function, and that's a function of a set of observations. And the optimization is done by changing the parameters of a model. So this may be a bit heavy if you just read it very quickly. So here's a little flow chart, what that means. We start refinement by supplying an initial model. We have our experimental data. So for cryo-EM, that would be the map. And we have a priori knowledge. We use all this information to score the model against the map. And then we modify the model parameters in order to get a better score. And then we possibly repeat this. And at the end, we get hopefully a better model. So let's go back to that definition. So there are a few keywords which are here highlighted in red. These are the parameters of a model, the observations, the function, and the optimization. So I will go through the first three keywords here. I will not talk about optimization because we don't have so much time. So let's start with the parameters of the model. So the model is a mathematical description of the molecule, which we use to perform our experiment. And so the molecule is composed of atoms. So it is the first set of parameters we can look at are the atomic parameters. So here is an illustration. So these two, in the bottom here, we have PDB string. So that's 2 lines from a model file, which show the properties of the C alpha atom in a proline residue in chain A in residue number 4. And one parameter to describe an atom is the coordinates. So these are 3 values to describe the position. Another parameter is the B factor that describes the local mobility. This can be either a single number, or in the case if the thermal motion is isotropic, or it can be 6 numbers if the motion is anisotropic. And finally, we have the occupancy, which describes large-scale disorder. So apart from the atomic parameters, we can have other parameters. So for example, in the case of crystallography, we can have bulk solvent, anisotropy, or twinning. And for both cryo-EM and crystallography, we have rigid body parameterization, or NCS, non-crystallographic symmetry. So, and there are various other possibilities to add parameterizations. So now we have an idea how to describe the molecule, and let's move on to the observations. So the observations are everything we know about the sample. So very simply, that would be the experimental data. So that's something which describes our sample. And then we have standardized stereochemistry. So for example, if our molecule is a protein, we know that the protein is composed of amino acids, and we have a pretty good idea how an amino acid looks like from other experiments, such as small molecule crystallography. So we know about bond lengths, bond angles, if there are planes, and so on. And we can use this information to guide refinement. And then we have other information, such as the sequence, or in crystallography, the unit cell parameters. So I want to go quickly again over the derived experimental data. So we've seen that in the previous talk. In crystallography, the interaction happens between the X-rays and the electron density in the sample, in the crystal. And this yields structure factor amplitudes. And structure factor amplitudes is a quantity which lives in reciprocal space. So it makes sense to perform refinement in reciprocal space. And in cryo-EM, the interaction is between the electrons and the electrostatic potential. And this yields a cryo-EM map or 3D reconstruction. And this is a quantity which is in real space. So it makes sense that we perform refinement in real space. So another aspect of the prior knowledge is the restraints. So as I mentioned already in the beginning, cryo-EM maps have typically quite low resolution, so between 3 and 5 angstrom. And here you can see how the resolution changes the shape of the density. So here I have a density map at 1 angstrom, at 2 angstrom, and at 3 angstrom. And we can see at 1 angstrom we have peaks at each individual atom. At 2 angstrom it gets more blurry, but we still have that hole here in the ring. And here on the right, at 3 angstrom, we have only one blob left. And we can imagine at 4 and 5, that gets even worse. So the lower the resolution, the less features we have in the map. And we need extra information to keep the correct geometry during refinement. So let's say if we only use the density to refine these atoms here at low resolution, it might happen that the peak of the density is actually not where the atoms are located, but probably in the center of this blob. So what happens then is that each atom will end up in the center of the blob, and it will not look like the correct geometry of the side chain. So for refinement, we will supply restraints, and the restraints will make sure that these atoms have correct bond lengths and angles and form a plane. So when we use restraints, we say that the model property is approximately equal to an ideal value, and this will add prior observed information. Okay, so the third component now for refinement, that's the target function. The target function will score how well the model explains the observations. And to do this, we can use least squares or maximum likelihood, or maybe other methods. And again, I want to discuss why it is very useful to have a cryo-EM target function in real space. So one reason is that the conversion from a map to structure factors may not be lossless. And I think it's very clear that when we deal with our experimental data, we do not want to lose any information when we do refinement. Then if we do refinement in real space, the calculations are faster. And this is important because we are dealing with very large structures. So some of them are really very, very big. And if the calculations are faster, we can get our results faster. And then we also want to have a quick turnaround when we do some development or model building. So if we want to, let's say, test a parameterization, we want to see if it works in maybe 2 hours or 20 minutes and not in 2 days. And then if we do perform refinement in real space, we can use local targets. So I want to highlight what that means. Let's say we have our protein, and let's say in refinement, we change the orientation of a side chain. So in reciprocal space refinement, if we change one side chain, this will affect all calculated structure factors. However, in real space, this will also only affect the calculated density locally. So in this area here, while if I'm here, probably this area will not feel if this side chain has moved. So let's have a look how the target function looks like in Phenix. So the target function is a sum of a database term and a restraints-based component. So the database term represents the model to data fit, and the second term here, the restraints-based term, incorporates a priori information about the model. So let's focus now on the database term. So one way to describe this term is to say we want to optimize the fit of the shape of the densities. So we want to fit the shape of the observed density, which we have from our experiment, and the model calculated density. So for example, we can use a least squares term. So this would translate to the integral over the square of the difference between the two densities. And now this is probably not very fast to do. It's maybe also a bit complicated to do. So probably we can do some approximations. And so I will not go into detail how this has been done, but you can read it in Pavel Afonin's paper about Phenix real space refine. So according to his approximations, we can simplify this least squares term to now from an integral to a sum over all atoms. And we have here the experimental density. So that means we will fit the atoms to the nearest peak in the experimental map. So if you compare this to our starting point, we wanted to optimize the shape of the densities, and now we will only fit the atoms to the peaks. So we have here like a simplification, but in the regime of, in the regime of the resolutions we have in cryo-EM, this is sufficient. So now let's look at the second term, the restraints-based term. This term itself is again a sum of different contributions. Let's say bond restraints, angle restraints, dihedral restraints, and then we have also some other restraints such as non-crystallographic symmetry, Ramachandran restraints, reference model restraints, and so on. They're usually expressed as a harmonic function. So for example, in the case of bond restraints, they are a sum over all bonded atoms. Weight terms the square between the difference between the distance we find in the model and the bond length we have in the ideal bond length. So at low resolution, what we have in cryo-EM, we have a set of many restraints. So again, we have covalent geometry, which we can restrain, but we can also restrain the side chain orientations, because at low resolution, it happens often that there's no or not sufficient density for side chains. So we can maybe assume that these side chains will adopt conformations found in higher resolution structures. And similarly, we can assume that the main chain will follow the Ramachandran distribution. We can also use the information of related structures. So let's say we want to refine a big complex, and if maybe one domain has been already determined with another method, we can use this knowledge of the structure of this domain for the complex. And also we can use internal symmetry. So let's say if our protein or a molecule is composed of different chains, which are all the same, then the copies of these chains are likely to have similar conformations. And then another restraint we have is secondary structure restraints. So here's an example which shows the refinement of a perfect alpha helix into a low resolution map. So here we have the helix, and this mesh shows the map. So it's very tubular, it's low resolution, and let's see what happens if we just let refinement go on. So our nice regular alpha helix now gets distorted. So if we don't use some additional information, then the model geometry will deteriorate. So in order to prevent this is to use secondary structure restraints. So we restrain the hydrogen bonds in secondary structure elements. So in helices and beta sheets, we have hydrogen bonds between peptide units. Shown here is these black lines. And in nucleic acids, we have base pairs and stacking pairs. So we can restrain the hydrogen bonds in base pairs, and we can keep these two groups here parallel. So now with all these tools at hand, we wanted to apply this. So we decided to re-refine some cryo-EM models deposited in the PDB. And we wanted to make these results available on a web page. So the motivation for this was to find bugs and to improve algorithms and protocols. We wanted to see how our tools perform, and also the tools are improving or evolving rapidly. So let's say if we do refinement today, it's most likely very different to how the refinement run went a year or 2 years ago. So here's a flowchart how we did this. So we took the model from the PDB and the map from the EMDB, because these two files are in different repositories. And then we had to do some initial curation. So as they are on different repositories, the validation is sometimes not perfect. So it might happen, for example, that map and model are actually not superposed. So if you just take map and model as they are deposited and open it in COOD or Chimera X, then they're not at the same place. And there can be also different information in the different files. So it happens also often enough that the resolution limit is different in the map file and in the model file. So once we have curated our set of map and models, we can move on. We get some initial model statistics, such as how many chains, how many residues, and most importantly, are there any ligands? If there are any ligands, we have to get CIF restraints. And then we can move on and get initial statistics. So that means we get model statistics, data statistics, and model versus data statistics. And then we can re-refine the model against the map. And then we get exactly the same set of statistics again, and we can compare. So the results from that re-refine project, they are available on the web. So you can access this web page at cci.lbl.gov/re-refine. You can search for maps or models, or you can just browse all the results in a table. So here's a screenshot how it looks like. We have a summary tab. Here is shown the model tab. So you have some information about the model geometry, such as Ramachandran plot and the content. And here are two examples of the re-refinement. The first one is about a structure determined at 3.7 angstrom. It has 4,000 residues. And here we have for different metrics, we have the values from the deposited structure and then from the re-refined structure. We can see that the correlation between the map and the model gets better a little bit. And we can see that the clash score decreases and we have less Ramachandran outliers or rotamer outliers. And then another example, this time it's a very big structure. It's a virus determined at 4.8 angstrom. Resolution, which has 31,000 residues. And here we have a similar correlation between the map and the model after refinement, but we have a better clash score, and we have fewer zero Ramachandran outliers and fewer rotamer outliers. So it's of course interesting to see before and after what happens, but actually the web page is also quite instructive to see some issues with some of the models. So here are two fun examples. So here is here's a model, and the map is superposed. You can see it a bit in transparent. And the resolution of this map, as indicated in the files, is 3.8 angstrom. However, I don't know, it's maybe not so visible from far, but this does not really look like a 3.8 angstrom map. So the resolution of this map, the apparent resolution, is much, much lower. And so we estimated the resolution in our statistics step, and this resolution is 7.5 angstrom. So it looks like the deposited map is probably not the map which has been used for refinement. And here's another example. So here again, we have a molecule, and the coloring here is according to the correlation coefficient between the map and the model. So here we have blue. Blue means good, we have a positive correlation, and red means bad. And here again, this surface shows the map. And now we immediately can see why there is such a bad correlation between the model and the map, because here we actually don't have any model. So the map, we don't have any map. So the map which has been deposited does not cover the entire model, associated model. So this is all I wanted to tell you about real space refinement. So as said, I'm in the Phenix, I work in the Phenix Project, and the Phenix Project is a collaboration between 4 different labs. One is in Lawrence Berkeley National Lab, that's where I'm working. And then we have Tom Terwilliger in Los Alamos, the Richardson Group in Duke University, and finally Randy Reed in Cambridge. So yeah, thanks again to the organizers for inviting me here, and I'd like to thank you for your attention.
Thank you, Dorothy. Do you have any questions? Randy at the front.
Dorothy, in that last example you showed where there was no model for the map, is it, if you look at the paper describing that, is it possible that focused refinement was used for those other domains?
I admit I did not look at the paper, but yeah, that's possible.
I guess this is a question to the PDB, but what validation are they using? Surely any validation procedure should notice that, shouldn't it?
Yeah, I don't know if the PDB actually ever sees the map when the model is deposited. I don't know about that. Maybe someone from the PDB can comment on that. Yeah. I don't see where.
Yes, so we're well aware of that. The deposition system for EMDB and PDB is the same one. They both come through one dep. We are just about to roll out EM validation reports, which would include analysis of this sort of thing, so volume inclusion, looking for where the map and model disagree. So this is available to depositors now, if you deposit a new structure, and will shortly be available to everybody in the full archive in both PDB and EMDB archives.
Okay, thank you.
Okay, one more question over here.
I have a question regarding to rotamer outliers. And I know that in many high-resolution X-ray structures, we can often see like 2 or 3% of rotamer outliers, but in many low-resolution quantum structures, rotamer outliers are extremely low, like even 0 point something or even 0%. Can you explain this observation? Or maybe I'm wrong, but I often see this in the statistics.
Let me just repeat that I make sure I understood. So you're saying that at higher resolution structures, you have more rotamer outliers than at low resolution?
Yes.
Is that correct?
Yes.
Yeah, so that might seem counterintuitive, but so the, let's say, mentality we apply is that at low resolution, there's most likely not enough data to really confirm that there is an outlier. So let's say if we have a Ramachandran outlier, maybe at high resolution, we really see the density for each individual atom, so we can confirm this is really an outlier. It's a genuine outlier. But maybe at low resolution, it might just happen because somehow the atoms move into that conformation which corresponds to an outlier. So we don't have, the data accuracy is not good enough to, how to say, justify that this is an outlier.
Yes, thank you. But I have this question because I saw this all the time from PDB. So I think to use rotamer outliers as a criteria to judge model quality regardless of the resolution, I think it's quite still problematic from my point of view. Thank you.
Mm-hmm.
Okay, if there are no more questions, let's thank Dorothee once again.